The color of urine is an indication of how much water is present in your system. For instance, if the urine is a pale yellow color, this shows that you are well hydrated. It means your cells have sufficient amount of water. But if the yellow turns any brighter, then it shows that you are dehydrated and that you should drink some water. However, if the color turns brownish, then this shows you are extremely dehydrated and you must increase your water intake. Otherwise, you are going to suffer from kidney failure. So in today's Bio World, I'm going to talk about how the kidney forms urine. Urine formation involves three processes, which are ultrafiltration, reabsorption, and secretion. But before that, let's discuss the importance of urine formation. We produce urine for two purposes. The first, as an excretion mechanism. Through urine, we can excrete metabolic waste products. For example, when we have excess amino acids, we can carry out deamination and ornithine cycle to produce urea, which we then excrete through the urine. The second purpose of urine formation is as a regulation mechanism. Through urination, we can regulate the chemical compositions of our body fluids by removing excess substances such as salt. Or we can regulate the water content in our body through homeostasis of body fluids. And we can also regulate the pH of our body fluids through the production of urine. So before we go into the details of how urine is formed, let's study the organ that is responsible for urine formation, that is the kidney. We have a pair of kidneys. The kidneys receive oxygenated blood from the dorsal aorta, which branches into the renal artery, and the deoxygenated blood from the kidney is transported via the renal vein up the vena cava. Besides the blood vessels, the kidneys also have a tube called the ureter. The ureter transports the urine that is formed by the kidney to the bladder. Now, if we look at the interior of the kidney, we can see the kidney has the outermost layer, which we call the capsule. The second layer below the capsule is the cortex, and the innermost layer of the kidney is called the medulla. The medulla is arranged in a set of triangle-like structures which we call the pyramid. Each of the pyramid will channel the urine that is formed into this region here called the pelvis. Then the urine will accumulate in the pelvis and flow along the ureter to accumulate in the bladder. This is the nephron, which is responsible for urine formation in the kidney. Here you have a ball of blood capillaries called glomerulus. This glomerulus receives its blood supply from the renal artery. The glomerulus then is encapsulated inside a cup-like structure called the Bowmans capsule, and from there the uriniferous tubule develops. The uriniferous tubule is made up of the proximal convoluted tubule, which is closest to the Bowmans capsule, the loop of Henle, and the distal convoluted tubule that is furthest away from the Bowmans capsule. All the distal convoluted tubules of the different nephrons will connect to the collecting duct. The glomerulus also extends to form a network of blood capillaries, where the blood capillary that encircles the proximal convoluted tubule and the distal convoluted tubule is called the peritubular capillary, whereas the blood capillaries that are surrounding the loop of Henle are called vasa recta. Eventually, the blood capillary will join the renal vein. The whole structure of nephron actually crosses between the cortex and the medulla. The glomerulus, the Bowman's capsule, the proximal convoluted tubule, and the distal convoluted tubule are positioned in the cortex layer of the kidney, whereas the loop of Henle will extend into the medulla layer of the kidney. As mentioned earlier, urine formation involves three processes. The first is ultrafiltration, which occurs between the glomerulus and the Bowman's capsule. The second is selective reabsorption, which happens firstly at the proximal convoluted tubule and secondly at the loop of Henle. This is followed with secretion, which occurs at the distal convoluted tubule. The urine that is formed will be channeled to the collecting duct to be excreted out of the kidney. So let's begin with ultrafiltration. The glomerulus and the Bowman's capsule are adapted for the process of ultrafiltration. We'll first look at the adaptation in the glomerulus. The glomerulus have high hydrostatic pressure due to the difference in the diameter between the afferent arteriole and the efferent arteriole. The afferent arteriole transports blood into the glomerulus, while the efferent arteriole transports blood out of the glomerulus. Since the diameter of the afferent arteriole is large, there will be a huge volume of blood entering the glomerulus. But the diameter of the efferent arteriole is smaller. That means less blood is exiting the glomerulus. The difference in the amount of blood entering and exiting the glomerulus develops into a high hydrostatic pressure. The molecules inside the blood are now being pushed onto the wall of the glomerulus. Now, the second adaptation is in the wall of the glomerulus. The blood capillary that make the glomerulus have semi-permeable walls due to the fine pores. So this means the molecules do not diffuse across the membrane. Instead, they are being pushed through the fine pores. And since the pores are small, only small molecules are permeable. The larger molecules like the red blood cell and the plasma proteins are trapped inside the glomerulus. Now, it's not only the glomerulus that is adapted. Let's look at the adaptation in the Bowman's capsule. Adaptation in the Bowman's capsule is related to the inner wall of the Bowman's capsule. The inner wall of the Bowman's capsule is made up of a specialized group of cells called the podocytes. The podocytes have finger-like extensions that wrap themselves around the capillaries. The podocytes increase surface area to enable efficient ultrafiltration to occur. Now let's combine all three adaptations together to understand ultrafiltration better. The high hydrostatic pressure in the glomerulus causes ultrafiltration to occur. Ultrafiltration is when smaller molecules such as water, salts, glucose, amino acids, and urea are forced out of the glomerulus through the tiny pores on the glomerular wall. Meanwhile, the larger molecules such as red blood cells, platelets, and plasma proteins remain trapped inside the glomerulus. These smaller molecules will then move down into the Bowman's capsule due to the presence of the podocytes on the inner wall of the Bowman's capsule to form the glomerular filtrate. The content of the glomerular filtrate is similar to that of the blood plasma. Only thing, the larger molecules are not present in the glomerular filtrate. Meanwhile, the blood that remains in the glomerulus will have less of the smaller molecules. The blood that remains in the glomerulus after ultrafiltration will be transported out to the peritubular capillaries. and the glomerular filtrate will travel along the uriniferous tubule to form urine. But there's a problem with ultrafiltration. The problem lies in the fact that ultrafiltration is non-selective. So you see, the glomerular filtrate now has both waste as well as useful substances mixed together. The waste, like urea, is to be excreted, but useful substances such as glucose cannot be excreted. They are important for the body. So, the solution has to be in the next two steps of urine formation, that is, reabsorption of the useful substances such as glucose and secretion of waste substances such as urea. So now we move to the process of selective reabsorption. Selective reabsorption occurs in two locations, firstly at the proximal convoluted tubule and secondly at the loop of Henle. The proximal convoluted tubule has a hollow passage to transport the glomerular filtrate. This passage is called the lumen. The blood capillaries that surround the proximal convoluted tubule are the peritubular capillaries, and the space in between is known as the interstitial space. Just like how the glomerulus and the Bowmans capsule were adapted for ultrafiltration, the proximal convoluted tubule is also adapted for selective reabsorption. The walls of the proximal convoluted tubule are made up of simple cuboidal epithelial cells. These cells have many microvilli on its surface. So, this surface is at times called the brush border. The microvilli will face the inner lumen of the proximal convoluted tubule, and its function is to increase surface area so that reabsorption can happen at a higher rate. To increase the rate of reabsorption, the simple cuboidal epithelial cells can also carry out pinocytosis. So, during fluid endocytosis, pinocytotic vesicles will form. These vesicles will be able to transport useful substances such as glucose, amino acids, and even salt to the opposite end of the cell. On the opposite end of the cell, there are many channel and carrier proteins to enable facilitated diffusion and active transport of the useful molecules from the lumen of the proximal convoluted tubule into the peritubular capillary. As you can see, since active transport is involved, naturally these cells will have a large amount of mitochondria to generate ATP. Let's visualize selective reabsorption in the proximal convoluted tubule. The lumen of the proximal convoluted tubule contains glomerular filtrate. The glomerular filtrate will have useful molecules such as the amino acids, glucose, sodium ion, and water. It will also contain waste substance such as urea. Now, the objective of selective reabsorption is to reabsorb useful substances from the glomerular filtrate back into the blood found in the peritubular capillaries. To help in the process of reabsorption, we have the simple cuboidal epithelial cells. These cells have a larger surface area due to the presence of the microvilli. So, useful substances like amino acids, glucose, and salt can be reabsorbed efficiently into the cells. From the cells, these substances are transported into the peritubular capillaries either by facilitated diffusion or active transport. The amino acids and glucose are transported by active transport with the help of carrier proteins and ATP generated by the mitochondria. You can notice that the glomerular filtrate will have no amino acids or glucose anymore. This is used as an indication in urine tests. A healthy person will show no presence of glucose and amino acid. If glucose is detected in the urine, that indicates the person has diabetes mellitus. If amino acid is detected in the urine, that could be a sign of kidney failure. However, you see, sodium ions are only transported by facilitated diffusion. So that is why you find the glomerular filtrate will still have some sodium present. Now, the presence of the amino acid, glucose, and sodium ions in the peritubular capillaries cause the osmotic concentration of the blood in the peritubular capillaries to increase. So this will cause water to diffuse by osmosis into the peritubular capillaries. So the glomerular filtrate now will only contain sodium ions, water, as well as urea. So this is now called filtrate. The filtrate produced from the selective reabsorption at the proximal convoluted tubule will be transported down to the loop of Henle for the second stage of selective reabsorption. But to hear about the second stage of selective reabsorption, you'll have to wait for my video on urine formation part 2. Until then, bye-bye.